Okay, so welcome to this next video in the playlist on lipid modifications of proteins. In this video, what we're going to do is discuss caged inositol trisphosphate. Okay, so what we're going to do is we're going to start off with an example problem which we could use inositol 1,4,5 trisphosphate, which is caged, to solve, basically, or at least get some information from. Okay, and inositol 1,4,5 trisphosphate and inositol trisphosphate, they're the same thing. Inositol trisphosphate is just the shorter version of inositol 1,4,5 trisphosphate. Okay, right, so I'm going to start off with an example problem, basically, and we'll then talk about what the theory is that for this problem, basically, and then we'll see how we can use caged IP3 to give evidence that backs up this theory. Okay, right, so basically the example that we're going to give is based on a paper, okay, and I'll give you the title of this paper here in case you want to go away and read it. Okay, so the title of this paper is Stable Expression and Function of the Inositol 1,4,5 Trisphosphate Receptor Requires Palmitoylation by a DHHC6 Selenoprotein K complex. Okay, now if you type that into Google, hopefully you'll at least be able to get up the abstract for the paper from PubMed or something, and hopefully you'll be able to get access to the full text. Okay, right, if you should want to read that. Right, so let's now talk about the example problem, and then we'll talk about caged IP3. Okay, so the example problem, basically what they did in this paper is they knocked out a protein known as selenoprotein K. Okay, now selenoprotein K is a protein that is within the membrane of the endoplasmic reticulum. So let me just draw a picture of a cell here. Okay, so here's our cell, here's the nucleus, and a very important intracellular organelle that is membrane bound is the endoplasmic reticulum. Okay, so this is the endoplasmic reticulum, which is often just abbreviated to ER for short. E for endoplasmic, R for reticulum. Okay, right. So, the endoplasmic reticulum is surrounded by a lipid bilayer, which is just like the plasma membrane lipid bilayer. Okay, and in this membrane of the endoplasmic reticulum, let me draw the endoplasmic reticulum here, there is this protein known as selenoprotein K. And for short, selenoprotein K is often abbreviated to cell for selenoprotein, and then K for K. So SELK is the shorthand for selenoprotein K. Okay, now you might be wondering, why is it called selenoprotein? Well, basically, selenoproteins are a huge, great family of proteins, and the characteristic feature that all of them have is that they contain a selenocysteine, at least one selenocysteine amino acid. So let me show you the structure of a selenocysteine amino acid residue. Okay, so I won't draw the pure amino acid, I'll draw it as though it's within a polypeptide. Okay, so here is the amino group, which will be bound to the carboxylic acid group of the amino acid that was before it. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, so this is the amino acid core structure that we've shown so far. And then the R group of selenocysteine has a methylene group, then with a with a special group, basically. So if we think about cysteine, the R group of cysteine would be a methylene group with then a thiol group sticking off it. Okay, so with a sulfur atom and then a hydrogen atom off that. So I might draw this cleverly. Okay, so it would have a sulfur atom, then with a hydrogen atom bound off it. Now in selenocysteine, this sulfur atom is going to be replaced by a selenium atom, which is why I left that space there for the E. Okay, so Se is short for the atom selenium. And selenium is in the same group of the periodic table as sulfur. So basically, if you look at group 6 of the periodic table, okay, right at the top there is oxygen. Underneath it there is sulfur, and then underneath sulfur there is selenium. Okay, so sulfur and selenium have very similar chemical properties, and both of them then have similar chemical properties to oxygen. Okay, so in selenocysteine, what you have done is replace the sulfur in the thiol group with a selenium atom. Okay, and selenoproteins all contain these selenocysteine residues. So that is why it's called a selenoprotein, basically. And this is a specific example of a selenoprotein. It's selenoprotein K. Okay, so here it is. Now, basically, what these experimenters found, is that if you knocked out selenoprotein K, so if you either knocked it out genetically, maybe by an siRNA directed against the mRNA of the selenoprotein K, or if you made the cell deficient in selenium, which meant that you couldn't produce the selenocysteine amino acids, then that would also knock out the selenoprotein K. Okay, what they found is that if you then stimulate the phosphoinositide pathway, which leads to the production of IP3, which then activates IP3 receptors in the ER membrane and then causes the release of calcium from the ER. Okay, so IP3 will act on these IP3 receptors here, which will then cause the release of calcium from the ER into the cytoplasm. And it's not IP3 that directly causes the release of calcium, but it primes the receptor so that it can then release the calcium. But for a useful simplification, we can think of IP3 causing the opening of the IP3 receptor here. Okay, now basically they found that if you stimulate the phosphoinositide pathway that generates IP3, and then look at how much calcium you get released from the ER, they found that the calcium that was released from the ER in in cells that had selenoprotein K knocked out, they found that the calcium flux was smaller, basically. So without selenoprotein K, the amount of calcium that's released from the ER when you stimulate the phosphoinositide pathway will be reduced, basically. Okay, and their interpretation for this is that selenoprotein K forms a complex with another enzyme in the membrane of the ER. So let me show you this other enzyme. So this other enzyme is going to be an enzyme known as DHHC6. Okay, so the DHHC family of enzymes is a large family of enzymes. There are 23 different DHHC enzymes. DHHC6 is a specific example that is present within the ER membrane. Okay, and all of these enzymes are involved in the palmitoylation of proteins. So, let me now show you the proposed complex of DHHC6 with selenoprotein K, and then we'll discuss the palmitoylation of these IP3 receptors, that is, the interpreted reason for this result, and then we'll discuss how caged IP3 can be used to back this result up. Okay, so, let's start with the DHHC6 enzyme. Okay, so I'm going to show you its membrane-spanning topology. Okay, so we start off with the amino terminus of the DHHC6 protein. Then it has a membrane-spanning alpha helix, a second membrane-spanning alpha helix. Okay, and I should just stress, this is the cytoplasmic side here of the membrane, and this is the lumen of the ER, okay? So this is inside of the ER here, and this is outside of the ER in the cytoplasm of the cell, basically, here. Okay, right. Now, in between the second and third membrane-spanning alpha helices, you have a very important region, which I'll come back to. Then you have your third membrane-spanning alpha helix, your fourth, another very important domain here, and then your carboxylic acid terminus of the protein here. Right, so what are these two extremely important domains? So this one here, between the second and third membrane-spanning alpha helices, this is what is known as the CRD for short. And in full, the CRD stands for the cysteine-rich domain. Okay, so this is a stretch of the polypeptide which is rich in cysteine residues. Okay, and specifically within this cysteine-rich domain, there is a very important sequence of amino acids, okay? So this is a long sequence of amino acids, but there are 4 amino acids in here which are always conserved. So all of the DHHC enzymes have a cysteine-rich domain, and in these cysteine-rich domains there are 4 amino acids that you can always find present, basically, okay? And this is a little sequence that goes DHHC. Okay, so within the cysteine-rich domain, there is a sequence of 4 amino acids that is always within that cysteine-rich domain. And the cysteine-rich domain is going to be extremely important for the catalysis of the palmitoylation of proteins, which we'll discuss in a moment. Okay, and this DHHC portion is extremely important for that palmitoylation. Okay, so this is obviously why these enzymes are called the DHHC enzymes, and to understand what this means you need to know your single letter amino acid codes. So D is the single letter amino acid code for aspartic acid. Okay, so I'll just show you the R groups of these amino acids. I won't draw the full amino acid structure, so I'll just show the R group. So basically, I'll omit the core structure, I'll omit the amino group, the alpha carbon, and the carboxylic acid. I'll just show you this R group that varies for the different amino acids. Okay, so the R group for aspartic acid is basically a 2-carbon carboxylic acid group, like so. So a methylene group with a carboxylic acid group off it, like that. Okay, this next amino acid along, at the second and third amino acids here, they are both H's, and H is the single letter amino acid code for histidine residues. Okay, so the R group of histidine amino acids consists of a methylene group with then an imidazole ring. So let me show you the structure of an imidazole ring. So imidazole rings are 5-membered rings where 3 of the members are carbons and 2 of the members are nitrogens, like so. And you have a double bond here and a double bond here, and then you saturate everything else with hydrogens. So the hydrogen here, a hydrogen here, and a hydrogen here. And this ring is what's known as an imidazole ring. Okay, and the reason it's called this is that this double bond between a carbon atom and a nitrogen atom, whenever you have a double bond between a carbon atom and a nitrogen atom, this is known as an imide bond. Okay, so that's where the imide comes from. Okay, also the fact that you have 2 nitrogens, whenever you see anything with azo in, that means pertaining to nitrogen, so it's about nitrogen. Now we have 2 nitrogens, so dazole there kind of reflects the fact that you have 2 nitrogens, because it's kind of like diazo, 2 nitrogens. So imidazole, that's the kind of logic behind this name here. Okay, so that's the R group of a histidine amino acid. So you have 2 of those, followed by the final amino acid, which is a cysteine amino acid. Okay, now what is the R group of a cysteine amino acid? Well, we've already covered this, but we'll write it out again. So the R group of a cysteine amino acid is a methylene group, then with a thiol group off it, like so. Okay, so basically, this enzyme is involved in palmitoylating other proteins, basically, and we'll discuss that in just a moment after I've discussed the assertion that this paper makes, which is that the DHHC6 enzyme is complexed with the selenoprotein K. So let's now discuss this other important domain over here. So this domain is what's known as an SH3 domain, which stands for Sark homology free. Okay, so this is a Sark homology free domain. Okay, so Sark, remember, stands for steroid receptor coactivator, and it's involved in glucocorticoid pathways. So this is a domain that is present within steroid receptor coactivator, so it's homologous to a domain in steroid receptor coactivator, and it's the 3rd homologous domain. So basically, SH3 domains are domains that you have within many different proteins. It's just a conserved structure that many different proteins have within them. Okay, now basically, the selenoprotein K also has an SH3 domain on its cytoplasmic side. So maybe I'll draw this here. So there might be some SH3 domain here. Okay, and the SH3 domain of selenoprotein K can associate with the SH3 domain of DHHC6. At least this is what this paper asserts. This is still putative. This paper only came out in October 2014. Okay, and the claim is that the DHHC6 enzymes will associate with the selenoprotein K to form this DHHC6 selenoprotein K complex, and that this complex is now going to palmitoylate the IP3 receptor. Okay, right, so let me now discuss palmitoylation with you a little bit, and then we'll discuss the structure of the IP3 receptor, and then we'll discuss caged IP3 and what this can be used to show us about this. Okay, so palmitoylation, specifically we're going to look at S-palmitoylation of proteins. So there is also a form of palmitoylation called N-palmitoylation, which is much, much rarer than S-palmitoylation. Okay, right, so let's start off with the structure of a palmitic acid molecule, because this is the star of palmitoylation. Okay, so palmitic acid is the old biochemist's name for a molecule that chemists would now call hexadecanoic acid. Okay, and although hexadecanoic acid is longer, it is a useful name because it tells you exactly what this molecule is now. It's a 16-carbon carboxylic acid where the whole structure is saturated, basically. So let's draw this out. So we start with our carboxylic acid group here, and now we need to have 15 other carbons coming off this to take us up to 16. So we're going to have 14 methylene groups, and because I don't want to draw out 14 methylene groups, I'll draw 1, brackets around it, and then put a subscript 14 like so. That's a useful trick for avoiding having to do a lot of painful work. Okay, then finally, right at the end, you have a methyl group coming off this, and hopefully you'll agree that that takes us now up to 16 carbons. We have this one and this one, that's 2, and the 14 that are in the middle as methylene groups, that takes us up to 16. Okay, now you don't directly add palmitic acid molecules onto proteins. Instead, you use a molecule known as palmitoyl-CoA. Okay, and CoA is short for coenzyme A, and this is the same coenzyme A that you probably know from the respiratory reactions, and the way that it is bound to palmitic acid is pretty much the same as it would be bound to acetyl in acetyl coenzyme A. Okay, so basically we're not going to draw out the full structure of coenzyme A. Instead, we're going to write CoA in a box, and that will do. There's only one functional group that is important for us, and that's a phyl group that comes off the coenzyme A. Now, I would encourage you to think of phyl groups as being analogous to alcohol groups, okay, because sulfur is in the same group of the periodic table as oxygen. So imagine for a moment that that sulfur is not a sulfur and that it's an oxygen instead, so this phyl group is no longer a phyl group but an alcohol group. Then we could react this alcohol group with the carboxylic acid group and form an ester link. Now what we do is we take the alcohol group off the carboxylic acid molecule, we take the hydrogen off the phyl group, off the alcohol group that we're pretending it is for the moment. We bind those together to make water, okay, so this would be a condensation reaction, and then we bind this carbon here, let's take it, to the sulfur atom, okay, so it would then end up bound to the, we're pretending this is an oxygen atom. It would bind to the oxygen atom, and that would be called an ester link. Now basically you can do the exact same reaction for phyl groups as you can do for alcohol groups. So you can bind this sulfur atom to this carbon, and it's no longer called an ester link, but it's called something analogous to that. It's called a thioester link, basically. So you can link coenzyme A to palmitic acid molecules by a thioester link, okay, and this produces you palmitoyl coenzyme A. Okay, and it's these palmitoyl coenzyme A molecules that you are going to use for S-palmitoylation of proteins. Okay, and we'll discuss S-palmitoylation of proteins in the next video.